try to, it'd probably be unethical. I can manipulate a mouse genetically, but it's very expensive and very slow. It's also very challenging, technically. This guy is really easy to manipulate genetically because you can literally just apply a chemical mutagen and then look for animals that are abnormal in the way that you want to study. So I'm going to show you an example of that in a second. So we just call that genetic malleability. That is, it's easy to modify the genetic code in this animal and trace where the change is and what it does. That's really hard to do. I mean, even studying a disease and looking at, let's say 30 people that have a disease and you trace their family lineage and you try to pin it down to one genetic change, that's hard to do. And it's hard to do it accurately and be certain that that's causing the problem. Whereas here, we can always be certain, because once we've nailed down what the change is, we can actually repair it. We can fix it ourselves again and show that that causes something. That's a really, really powerful scientific concept that I think most students, even in undergraduate education, really struggle with. This idea that, a genetic lesion, kind of what is it? What does it mean? What does it do? And the way I find easiest to understand is that one gene encodes one protein, and when you mutate that gene, you lose function, you change it. And the key to understanding that is being able to put it back in. That's the key, is to being able to put back in a copy that's correct. And that's proof, right? That's the proof that that gene causes that effect. Okay, and then the last one is what I just showed you up here, which is that because they're transparent, we can see inside their body with really not sophisticated technology, basically.
Mark, is this the same protein that we use to transform E. coli from jellyfish?
Yeah, it is. Yeah, it's green fluorescent protein. So there's all kinds of derivatives and variety for that. Another guy from C. elegans won the Nobel Prize for discovering that you can use GFP in that way that I just showed you. He shares it with the people who discovered and started manipulating the green fluorescent protein from jellyfish and using it to make different colors. So some of the more sophisticated things we will do is if I want to study two proteins in space and time, I can make one green and I can make one red. Now I can follow them both around, watch where they go, look at how they behave relative to one another. And that leads us into the concept of cell biology. That's essentially what cell biology is. It's studying the cell and its morphology and how the things in it relate to one another through a microscope, usually. That's generally the definition of cell biology. The point of this slide is these are the techniques one uses in C. elegans to study these questions. The primary one is genetics. That's the big arrows. That's why the organism was first used. That's its most powerful technology, is the ability to manipulate its genes. The other one that I would say is the second most common is cell biology, where just observing and watching what happens to molecules in the cell and structures in the cell and how they change in different animals that are mutated for certain genes, or just in a normal setting over time and development is very informative. And then the concept of biochemistry. Well, so what's biochemistry? Well, that is the ability to basically destroy and pull apart the molecular machinery inside the cell and see how it works. So my lab tends to primarily focus, so you notice the little arrow is the primary focus of my lab. So I'm a biochemist. And that's what we focus on in my lab mostly, is understanding the biomechanical machinery that drives how you make a synapse. So the proteins inside the cell that say, let's make one here, and let's make it in this way, in this time and space. And then we use these as more secondary measures. So in a traditional C. elegans lab, this would be the primary measure. This would be the secondary means of investigation, and then this would be something they may or may not do at all. Okay.
How about electrophysiology?
Well, that's not even up here, because that's also kind of in the realm of this one for biochemistry. It's used, but very rarely. So I think there's 3 people that can do it in the world, in their own labs successfully. So it's very difficult. That's growing because those people that are doing it, they reproduce and they create other little scientists that can do it too, and then before you know it, you have 10 labs that can do electrophysiology and C. elegans. So very powerful technology. So to explain, electrophysiology is when you have 2 neurons that are connected, okay? And you want to understand those chemical messengers that run across the membrane. Okay, so what actually happens is that the consequence of that is a voltage change. So the cell actually changes the flux of ions across its membrane and charges itself. That's how signals get sent. So once the synapse is fired, this big change in ion flux occurs in the cell receiving the signal, and then that pulses down its body of the cell, through its axon, and lets it fire to the next cell. Okay? You can measure that by taking a tiny microscopic needle, attaching it to the cell, and then basically ripping open a little hole in the cell. Now you can, watch, actually I shouldn't say it's ripping open, it's not really, it's more like sucking it in. And now you can measure flow across that. You can actually measure the ions moving across that membrane. And that tells you the cell's charge and voltage. And it's a direct readout of activation state and whether or not the cell is releasing neurotransmitters. Okay, that's a very complex idea that I didn't introduce, but since you asked it, we can talk about it.
I just have to stick up for the electrophysiology.
No, absolutely. That's like, I would say, so the difference between C. elegans and probably a mammalian neuroscientist is that a mammalian neuroscientist, most likely the primary needs of investigation would be electrophysiology. That's an extremely common technique in mammals. We didn't even have it in C. elegans until probably 10 years ago. It took a long time to develop and even do, mostly because the size of the animal and the limited access to the nervous system. So whereas in a rat you can dissect out the brain and boom, you have these enriched neurons, you can't do that in worms very easily. So we use a series of tricks to get at the neurons. So with the simplicity of the system there, it backfires on you a bit because you don't have compartmentalization the same way. So electrophys is much more challenging, but highly informative. I wouldn't want to knock electrophysiology.
No, that's just
That was just, the C. elegans focus is that, anyone really can do it. So when we talk about studying neuroscience and C. elegans, how do we do it? What are the types of questions we're interested in, right? What are the types of things you can learn about? Now, I'm not talking from a health perspective. Some of these are obviously related to health. Most of these can be spun in the context of health, depending upon how much knowledge you have about how you use the information. But let's just start with ideas, okay? So, movement. So I already kind of hinted at that. But the most obvious form of movement that we're going to learn about today is something called uncoordination, or UNC. It's the classic phenotype that was first described in the 70s when the first worms were mutated. And it's just a simple principle. Normally the animal moves, and there's varying degrees in which it will become paralyzed. And with a single genetic change, a single gene being mutated, basically. And that gives you information on how the animal moves. Because we can map and figure out where that change is, and then understand what the nature of the change is. And now you understand movement, basically, in this animal on a molecular level. Not on the level of observation, which I was already mentioning is really difficult. Now we're on a molecular level, okay? So this has sort of been the classic. UNC mutants are just sort of the classic of understanding neuroscience. This is how people first put together the basics of neuroscience. I'm trying to think of like some of the examples. Some of the first molecules that were known to underpin how synapses work basically were described in C. elegans using this approach. If you had an electrophysiology rig hooked up and you were measuring current flow, nobody understood some of the mechanisms behind how that even happens. A lot of those have become unraveled using these UNC mutants. It was essentially the molecule that causes, when mutated, a loss of function, if you want to call it that leads to these body movement changes. And when they figured out, oh, it's this gene, then they realized, oh, that exists in human beings. Oh, bang, or rats. Bang, there it is. That's what's controlling that in rats too. So that's how you move from the simple system, what seems like an almost stupid observation of a worm that moves and one that doesn't, to something highly sophisticated. Okay, so neurodegenerative disease. Lots of molecules are related to this, can be studied. But the neat thing is that people can actually create models of disease. I put Parkinson's up here. It's not the greatest example. Huntington's is a better example. So you can actually take a C. elegans worm, and you can engineer it so it expresses wild-type Huntington, or Huntington with a lot of repeats in it that's like people with the disease have. And guess what? You create an animal that's really sick, an animal that has nervous problems. And that allows you then to bring the power of genetics back to understand that. So now you've created a system in which you basically made a worm that's got Huntington's disease. And I already showed you all the similarities in structure. I didn't show you the molecular similarities, but they're key. So that means that one can assume that probably the mechanisms of how Huntington's is occurring, for example, are similar. And now one can investigate that by mutating the worm and actually looking for an animal that gets healthy again. If you're able to find out how it gets better when you've created the disease, now you know what you need to look at in terms of helping someone who has it. It's a similar principle. And if anyone has any questions, we can talk about that more.
So on the trinucleotide repeat sequence, if you give it that extended trinucleotide, it's faster?
No, it actually paralyzes it. It actually causes loss of nervous function, and that leads to paralysis. So it actually mimics Huntington's even on the level of behavior in the whole organism, because the paralyzed worm is much like behaviorally, much like a paralyzed human being. So the similarities run very deep. Sometimes you will get that, where you see something unexpected, it could have a different consequence in worms. But generally, they tend to mimic pretty closely. So like with Alzheimer's, people express certain proteins that are associated, like it's a big controversy about what actually causes Alzheimer's. There's lots of things people can observe happen. No one really knows the cause. It's still a deep mystery, which is why we understand the disease so poorly when you really think about it. We know what causes HIV. We know the virus. We know how the virus works. We know the virus target. We don't know anything like that about Alzheimer's disease. We know it's a complex trait. Genetically, that means it's not caused by a single gene. Huntington's disease, I don't have it up there, is caused by a single gene being altered. Alzheimer's, Parkinson's, not single gene traits. They're complex traits, which means understanding them becomes very difficult. In worms, people have created models where they take some of the proteins that have been implicated to cause Alzheimer's disease that may not even exist in C. elegans. They put them in the animal artificially, and you get neurodegeneration. You get a model of Alzheimer's disease, and you can use the same principle to study how to fix it by looking for things that make it better. That's still pretty early in development, so I can't tell you a lot about what actually has come from that. So that's one of the most easy to convey links to medical science, along with this one. How neurons develop. So this is more like what I study. So we can understand the molecules and genes. So gene encodes a molecule. I'll just say that. The genes and molecules that regulate these processes. And like I said, when we understand those, we really get a long way towards understanding what happens in a human being. The mechanism is largely similar. All we have to do is magnify it, or amplify it, 1,000, 10,000-fold, and you have the same things happening. And what we find in the human brain, you don't find as much in stuff like C. elegans or Drosophila flies, fruit flies, is compartmentalization, which becomes the next big evolutionary step, which is as you get a bigger brain, you need to compartmentalize functions. Just like in a computer server, you can have a standalone computer that can do a lot of stuff, but soon you have to compartmentalize function, maybe linking 2 or 3 computers together and having them do one job, and then they're connected to 10 computers that are doing another job, and now you can synthesize information. Okay, so how do worms taste? So worms, C. elegans actually have a huge repertoire of taste receptors. The ability, they don't have a tongue, they actually have holes in their nose, and chemicals, whether they're volatile, if they're volatile, like a solvent or something, it can just actually diffuse right into the animal's body, up in the nose, and receptors sense the chemicals. They can sense aqueous water environments, and they have huge capacity to sense taste, basically, or smell, you call it, on a big scale. And I think that's one that I'll show you about for high school students, because that's one everybody can relate to. I can smell, I can taste, worms can smell, they can taste, and we can monitor that. Touch. C. elegans senses touch. When you hit the animal, essentially, when you poke it, it responds, it runs away. It senses pain and touch. So a lot of people, you think like, oh wow, this tiny little critter running around in the dirt. It actually has a pretty sophisticated touch system. It's not anything close to what we have, but it's a whole series of specialized neurons in its body that sense touch. And the last one here that I think is really neat is learning. So people have found that C. elegans actually can take information in and learn. It's called sensitization. So essentially, if I tap a worm, okay, if I give it an earthquake to its environment, it senses that and it runs around. Okay? And if you keep giving it earthquakes all the time, it actually learns that there's too much information coming in and it starts ignoring that. It's learning that its environment is really wobbly and shaking a lot, and it dampens down its touch sensitivity and stops responding. And that's a form of learning, and something that's very interesting to study. So now you think you have something simple, and you do, but now you just invoked a concept that has gigantic evolutionary implications, right? Because if you buy into the computer model I just gave you, that means that potentially the basics of learning on a molecular level may be intact in worms and may be highly relevant to human beings. So you can get a lot of insight that way. Okay.